Good evening, and thank you everyone for joining us at the Spaulding New England Regional Spinal Cord Injury Center Knowledge and Motion Program. Welcome also to our webcast attendees from 28 states from the US and countries such as Canada, Japan, Mexico, Nigeria, Pakistan, and the UK. Welcome everyone. My name is Dr. Sherry Blauette. I serve as a Director of Dissemination and Knowledge Translation for our spinal cord injury model system. We also are directed by Dr. Ross Safant and Dr. Andy Taylor. So this evening, we are honored to welcome Dr. Amanda Zimmerman to deliver our Knowledge in Motion lecture. She is a senior scientist at Axonis Therapeutics, a spinal cord injury-focused startup in the Boston area. Dr. Zimmerman received her bachelor's in biomedical engineering from Duke and her PhD in bioengineering from the Georgia Institute of Technology. As a graduate student at Emory, she worked on research of spinal cord circuits that control nerves that regulate the body's involuntary actions. In her postdoctoral research at Harvard, Dr. Zimmerman examined ways that a human body sorts out the signals coming from the environment and within themselves to allow them to feel and touch after such an injury. Before joining Axonis Therapeutics, she worked at the Christopher and Dana Reeve Foundation, focusing on identifying promising areas of spinal cord injury research. So welcome, Dr. Zimmerman. We're honored to have you with us today and look forward to hearing your presentation. And without further ado, I'll turn it over to you.
Hi. Thank you so much, Sherry, for that introduction. Let me just share my screen and we can begin. All right, so as Sherry mentioned, I'm a senior scientist now at Axonis Therapeutics. And I guess the term signal to noise ratio is a pretty common term in engineering. I apologize if it's not as common in your field of interest, but I think it's a reasonable framework to understand sort of the research area that we're working on. So as most people here know, the majority of spinal cord injuries are incomplete. About 3/4 of them consist of people who have either incomplete tetraplegia or paraplegia. But in the quarter of remaining injuries that are clinically complete, postmortem analysis of spinal cords from some individuals gives us an indication that most individuals that have a clinically complete injuries still have some amount of spared tissue that crosses the lesion site. So that leads us to one big question. How does anatomically incomplete injury result in a complete paralysis? More specifically, why does the remaining tissue not do very much? And is there a way we can reactivate it? Some therapies that many here might be familiar with, from epidural stimulation to intense rehab training to intermittent hypoxia, all get at this underlying question. How do we get more out of what remains? In order to understand why spared tissue is dormant, we need to understand a little bit about the function of the spinal cord to begin with. So we often think of the spinal cord as being a relay for information, but in actuality, it's a lot more complex than that. So here is a diagram from a recent review on circuits in the spinal cord that are responsible for processing touch and pain information. Now, I blotted out most of the names of the neurons because you don't actually need to know that to get this point. All of the neurons that are yellow or black or brown or pink or light blue all act as modulators. They modulate the signal coming into the central nervous system on its way to either elicit a movement or to reach the brain. And similarly, complex circuits modulate descending commands. And some even have the ability to generate patterns of activity all on their own, such as circuits responsible for locomotion generation. But how do these modulatory neurons work? There are two types of modulatory neurons in the spinal cord. There are those who help to propagate the signal, and there are those who help to inhibit it. Why might we want neurons that inhibit signals? I'll give you an analogy. I have 2 kids under 6. On any given day, I'm trying to convey a number of messages to them. And sometimes when they can hear me, they listen. Yet what happens when they're trying to hear me in, say, a crowded subway station? How do I get them to receive the messages that I'm trying to give? I could give my kids hearing aids, but that would only amplify the noise in addition to my voice. I could try a loudspeaker, or I could try to get everyone else in the subway terminal to quiet down. Your spinal cord is kind of like this crowded terminal. Neurons within it are responsible for trying to get the non-salient cues to quiet down so that you can pay attention to what's relevant. Another way to look at this, the spinal cord and most of the nervous system utilizes targeted inhibition. Quiet the noise so we can hear the signal. So when, say, we want to move our arm, a gauntlet of excitatory or go neurons propagate the signal, while inhibitory neurons, or stop neurons, seek to dampen the noise, or the signal is not particularly relevant to the task at hand. We can see this also in the opposite direction. At any given moment, our bodies receive a barrage of sensory information. For those currently sitting in a chair, there are neurons in your back, in your thighs, sometimes in your neck if you're leaning way back, that are constantly telling your nervous system, hey, there's a chair here. And while that information is relevant for your postural muscles to make sure you don't fall off of the chair, it's less relevant for you to pay attention to. Your nervous system will filter out that information before it reaches your brain, while allowing salient information, say the touch of a loved one, to get your attention. Yet what happens to inhibition in the spinal cord after injury? After a spinal cord injury, we know that there are weaker signals coming down from the brain. And we might hypothesize that there might be overactivity in inhibitory neurons that makes the signal seem even weaker, resulting in no or little movement in muscles. Yet on the flip side of the coin, many people living with spinal cord injury have chronic pain. This is often associated with hypersensitivity to heat and touch, and sometimes generates spontaneous activity on its own. Researchers think this is at least in part due to a loss of inhibition of pain circuits, causing excess pain drive to reach the brain. So hopefully by now, I've convinced you that inhibition is both useful for regulating the noise and allowing salient signals to reach your brain or the relevant muscles, and that dysregulated inhibition might contribute to some of the problems we see after a spinal cord injury. How does inhibition works? I know this is a weedsy slide, but bear with me for a second. Instead of stop and go neurons, neurons communicate with each other by way of neurotransmitters, chemicals released of either the excitatory or the inhibitory flavor. And the predominant form of inhibition in the spinal cord is through neurotransmitter-gated ion channels. This means then when the inhibitory neurotransmitter GABA is released from a neuron, it binds to a receptor on a neighboring neuron, and it opens an ion channel. The ion channel doesn't care which direction ions flow. That depends on the concentration within the cell and outside of it. In most neurons in the adult nervous system, there's a relatively low concentration of chloride in the cell and a much higher concentration out. And so when GABA binds to a GABA receptor, chloride flows into the cell, hyperpolarizing it. And that makes it less likely to be excited by other signals. Yet there is an ion pump, KCC2, that is responsible for pumping chloride out of the cell. And together with KCC1, these two pumps are responsible for making sure that levels of chloride within a neuron are meeting a set level. So as long as KCC2 is doing its job, every time an inhibitory neuron releases GABA, it leads to a hyperpolarization of the neuron and makes it less excited. Well, but what happens with injury? Well, it turns out this particular ion pump, KCC2, is downregulated after spinal cord injury. A number of researchers have shown that in pain circuits in the dorsal horn, there is less KCC2. What does this mean? It means there is an accumulation of chloride within the neurons, so that when GABA is released, instead of chloride flowing into the cell, it flows out. This means a loss of inhibition and sometimes even excitation, depending on how much KCC2 is downregulated. On the flip side, this also happens in motor neurons, such that motor neurons, the ones that innervate muscle, are also disinhibited. And the manifestation of this disinhibition is pain and spasticity. So if there is a decrease in KCC2, and therefore a loss of normal inhibition in both pain circuits and motor neurons, leading to spasticity, can enhancing KCC2 lead to improvements in either? One of our collaborators, Yves Stechonic at Laval University, screened for and found a couple drugs that actually enhance KCC2 function. And the first thing that his lab did was to look and see whether restoring KCC2 to near normal levels could improve chronic pain. As many of you might know, a lot of early preclinical research uses rodent models. And we can't just ask a rat or a mouse if something is painful. So we use an assay that is actually used clinically for humans as well, called Von Frey. And the idea is this: if you poke a paw or the palm of someone's hand with a light filament, humans and rodents don't really care. We might not even notice that you're doing it. But if you ramp up the force that you are poking someone's paw with, it'll get to a point that it becomes at least mildly uncomfortable. And rodents will tell you this by moving their paw. So we can actually measure hypersensitivity based on what force is necessary to get a rodent to move its paw. And our colleagues at Laval University did this. So they took a neuropathic pain model, in this case, spared nerve injury. Essentially, it's a surgically induced pain model. And we're able to show that after surgery, these rats were hypersensitive, as in they moved their paw to a much smaller force than in control. And yet they found that when they gave these rats this KCC2 enhancing drug, CLP-290, they were able to mitigate a lot of the hypersensitivity in both males and females. Well, what about spasticity? So luckily, before I got to speak with you guys tonight, a very interesting preprint came out of Marie-Pascale Coates' lab at Drexel University. And they were actually looking at spasticity in rats with a complete thoracic spinal cord injury and measuring it 5 weeks after injury. So spasticity is characterized by this velocity-dependent increase in the stretch reflex. And it manifests itself with symptoms such as increased muscle tone, hyperreflexia, and painful muscle contractions. It occurs in up to 75% of individuals living with a spinal cord injury. In this chronic rat model, their lab actually looked at hyperreflexia directly. So what they could do is stretch the leg of a rodent and measure the increased response in muscle. And so what you can see in the top trace here is that there is a lot of firing in the EMG recordings in response to muscle stretch. Interestingly, what they found is when they applied CLP-257, which was an earlier version of the drug I showed you last slide, they were able to depress a lot of this overexcitation. And in fact, they directly administered the drug to a window over the spinal cord, suggesting that the spinal cord is the locus of action for this. One additional way that we can measure both in humans and rats spasticity is to look at reflex modulation. And one of the most common ways of doing this is through what's known as an H reflex. So the idea of this is relatively simple. If you stimulate a peripheral nerve and you record the muscle that it innervates, you can measure both the direct muscle activation, called the M wave, as well as the sensory evoked reflex response, called the H reflex. And we know that there are circuits in the spinal cord that are capable of modulating reflexes. And one way to look at it is if you stimulate a bunch of times, you can actually see a depression in the reflex. Yet individuals with the spinal cord injury and rodents with a spinal cord injury see a loss of this modulation. So if you look at the blue trace here, what you'll see is as stimulus frequency is ramped up, there is not as much rate-dependent depression as you might expect in a control animal. Yet when they applied CLP-257 right over the spinal cord, they get a return to something that looks like pre-injury levels of modulation. Both of these metrics are good information to suggest that directly modulating KCC2 at the spinal cord can improve measures of spasticity in the rodent. So to summarize what I've told you so far, hopefully I've been able to convince you that inhibition is necessary for signals to meaningfully reach their target, whether that's the brain, the spinal cord, or muscle. That KCC2 is an ion pump that's a key regulator of inhibition. KCC2 is decreased in the spinal cord after injury and can lead to a loss of inhibitory circuits, resulting in both pain and spasticity. And drugs that directly enhance KCC2 can help mitigate these two manifestations. So what about the question that we started with? Is there an over-inhibition of commands from the brain after spinal cord injury? Is inhibition dysregulated to such an extent that it impacts motor function? One way to look at this, what if impaired inhibition also contributes to the dormancy of the remaining tissue? We know that the spinal cord is capable of generating locomotor rhythms. So why are these not easily activated after injury? If there is spared tissue after injury, can we strengthen what remains to get more out of less? And last, what if there is impaired inhibition of motor commands? So our colleague Zhigang He at Children's Hospital actually set out to find out the answer to these questions. And what he did was look at a pretty severe injury model in mice. This is a staggered hemisection for those who are curious, that results in a pretty severe paralysis. So if you just look at the blue line in this plot, what you'll find is relatively low scores on this metric for measuring locomotion. And none of the mice had any ability to generate stepping patterns. And yet when they virally delivered KCC2 back to these mice, they're able to see vast improvement on this locomotion score. Specifically, up to 80% of these mice were able to generate some sort of stepping movement. They next looked to see whether they could replicate this with daily drug delivery. So the idea is take CLP-290, the drug that Yves Deconick's lab had first investigated, and deliver it every day starting a week after injury to a different group of mice and see whether they also might improve. And it turns out they were able to replicate a lot of the findings with viral delivery. Mice that received this drug were able to generate stepping patterns in 80% of the cases. Now, through some complicated mouse genetics that I'm not going to show you here, they were able to identify which neurons were underlying this change. Specifically, it was a loss of inhibition of the inhibitory neurons that was dysregulated. So you can think of it as the stop sign on the stop neuron, that when that stop sign is lost, essentially the stop neurons are overactivated, leading to an over-inhibition of an already weakened motor command, and thus no movement. Yet one of my favorite parts about this paper is they wanted to ask why. If there are no direct connections from the brain, because this staggered hemilesion essentially severs all connections from the brain to locomotor regions of the spinal cord, how is drug delivery or viral delivery of KCC2 resulting in STEP? Well, what if instead of causing regeneration through the injury, they were able to just increase the connections indirectly of a windy pathway instead of a highway through the injury? Instead of motor commands going straight, what if essentially they form a relay, and a strong relay of that? Well, one way to test this is to stimulate the motor cortex. You can do this both in humans and in rodents. And humans, it's usually less invasive through transcranial magnetic stimulation. But essentially, if you stimulate the motor cortex, you can cause a muscle contraction, depending on where in the cortex you stimulate. And you can measure that muscle contraction by looking at the EMG response. So the bottom trace in red is EMG, showing that, yes, stimulate motor cortex, get a muscle contraction. Well, what happened when they did this in the staggered hemilesion mice? Well, if we look just at the top trace, what you'll see is in the immediate aftermath of cortex stimulation, there wasn't any signal. But if you look in a much larger time window, you do see kind of a wimpy muscle response. Yet in mice who had viral delivery of KCC2 or enhanced KCC2 function via CLP290, this longer latency component was strengthened. And note, in the complete SCI model, there's no activation because there's no way to get the signal from the brain to the muscle. So what does this mean? Essentially, this means that KCC2 therapy, either viral delivery or with CLP290, was able to strengthen these indirect pathways. Another way to look at it, if we take my initial model, KCC2 enhancement can improve the signal to noise ratio from spared fibers after injury, both by re-inhibiting the stop neurons and by strengthening indirect roots. So to summarize the preclinical findings from multiple labs to date, we think KCC2 enhancement has 3 targets of interest for spinal cord injury. By enhancing KCC2, we can restore chloride homeostasis and restore inhibition in pain circuits, hopefully improving chronic pain after injury, restore inhibition of motor neurons, therefore reducing hyperreflexia and spasticity after injury, and restore inhibition of inhibitory neurons, therefore improving the signal to noise ratio of motor commands. And ideally, we do this by a drug instead of a virus because viruses have a fairly long FDA regulatory approval in order to get out into humans. And small molecules or drugs are a much more straightforward pathway. So most people in the spinal cord injury community know that there are always a lot of promising therapeutics in preclinical studies that don't always translate out into the clinic. So I wanted to take my last few minutes and outline what is the actual process to get from exciting preclinical findings into something that people can actually use. Question 1. Will your therapeutic actually work in other species? A rat is not a mouse, is not a human. So having some confidence that any therapeutic works in multiple species gives us a lot more confidence that it will work in humans. Second question, can we design a better drug? In drug development, there are a few specific questions biotech and pharmaceutical companies go after, even if you know your target of interest. First, we need a drug to be able to survive our gut. In drug development speak, that's bioavailability. Second, when it comes to a drug that we want to target the spinal cord or really anywhere in the central nervous system, we need the drug to cross the blood-brain barrier. And this is essentially termed CNS penetrance. Can the drug get to where we need it to go? Next, we need the dose of the drug that is efficacious to be far away from the dose of the drug that can cause harm, toxic effects. And so we need to look at both potency and toxicology and make sure that they're far away on the spectrum. The last question is something that I learned a lot about while I was at the Reeve Foundation. If we want to design good clinical trials, especially for spinal cord injury, we need to admit that the spinal cord injury community is heterogeneous. There are differences in injury severity and injury level and injury cause, length of time since injury, any number of comorbidities that are expressed. And why waste anyone's time on a therapeutic that won't help them? So I think this is an area that I'm hopeful that more and more spinal cord injury therapeutics especially look at. It's called subject stratification. Can we predict ahead of time that this particular therapy will be useful for some people more so than others? And with that, I just wanted to thank the team that's supporting us at Axonis to try to find answers to some of these questions. And I'll take any questions.
Wonderful. Thank you, Amanda. All right, great. So we've had several questions come in via the chat during your talk. Thanks so much. Really, really interesting work. And, thanks so much for presenting it in a way that's really translatable to people with spinal cord injury and their families, as well as people working in this area, either in terms of research or clinical care. So really appreciate that. So we'll just take it from the top in terms of what's come in via the chat. The basic research question, what is viral delivery?
Yeah, that's a great question. So one of the ways to deliver any protein, essentially anything large, is to target it by virus. So actually, some of the viruses used in research labs are ones that are adapted from viruses that infect our nervous system. So one of the viruses that my old lab used to use a lot is actually related to the herpes virus, adjusted to try to make it safer for use. But the idea is if you can infect neurons, you can get them to uptake whatever it is you're trying to deliver. And this is particularly of value when we don't want systemic delivery, but we want to target it only to specific areas of, say, the brain or the spinal cord or the eye, depending on what injury that you're looking at. But one of the more promising ways of viral delivery right now is through a backbone that's adeno-associated viruses. And these are actually relatively safe. They won't jump from neuron to neuron. They'll stay in whatever neuron that you inject them into. And there's now a lot of research to make them even safer. So I actually wouldn't rule out viral delivery of any therapeutic, especially if there are genes that you want to regulate for regenerative purposes for the future. It's just a slightly longer pathway because people are always very, very cautious about viral delivery.
Yeah, sure. Okay. I know it's actually probably a harder question to answer than people realize because it's so technical. So thank you. That was a great, great explanation. Steve from the audience is asking about a specific question. He says, in my case, over the last 10 years as a para from thoracic SCI, I've had improvement in sensation and more muscle function, but that seems to correlate with more spasticity and increased pain. Does that fit with some of this research in terms of what you're seeing with KCC2, or excuse me, yeah, KCC2?
Yeah, it's a great question. I think one part of your question is easier to answer than others. The linkage between pain and spasticity is actually quite common. And one of the things that other labs have actually seen is exercise therapy itself is actually able to change KCC2 levels. So one of the reasons that we think that exercise might actually help, or rehabilitation training might help those living with injury, is that it would actually help restore them to a more normal level and lead to improvements in pain and spasticity. That said, there are probably a lot of underlying competing causes, so it's hard to diagnose out that specific example, but there's definitely a linkage.
Yeah, that's really interesting. Great. Do we know why KCC2 levels drop after spinal cord injury?
No. That's a really good question, and the timeline of them dropping is also something that researchers are still figuring out. I'd say the spasticity field is actually probably the furthest along. The lab I mentioned at Drexel has actually done some work in rodents saying, okay, after about 4 weeks, they hit this like new plateau. That I'm not sure the people studying KCC2 in the dorsal horn, where you might have modulation of pain or motor commands, what the time course looks like. There is some hypotheses that it has to do with BDNF. For those who recognize that term, it's essentially a transcription factor that has a transient change in expression after injury. But I'd say the jury is still out. In preclinical models, we can mimic KCC2 regulation by messing with BDNF, but that's more of a way to experimentally manipulate it versus knowing exactly why.
Okay, interesting. Great. Next question. Is there a time window post-injury for this approach to potentially work? Like, is there an ideal or at some point where we think that the impact may kind of burn out?
That is a great question. We're very much between us and some of our collaborators trying to find the answer to that. I will say when Zhigang He's lab started the oral dosing, they purposely started at 7 days after injury because they wanted to not get involved in the acute phase after injury. And from what we know, KCC2 is dysregulated for multiple months after injury. So I don't see why, at the very least, there is an automatic no for why we shouldn't have a much larger window. We just can't say for sure that it will improve all three of these until we try it.
Okay, great. So questions are coming in like crazy here. So Amanda, I hope you've had your coffee. We'll keep going. The lecture scheduled till 8, so we'll keep going. Really great, robust discussion. And thanks very much for your generosity with your time and helping us to work through these questions. Clearly a lot of interest in this topic. One question is, do we know whether bowel and bladder can also be targeted for improvement with this therapy, or is it really thus far focused on motor function and pain and spasticity?
That is a great question. I don't know the answer to that, but I do think that there's a good likelihood that it might be. And so some of the studies of folks that I mentioned earlier are actually starting to measure bladder function for that reason. But the short answer is I don't know yet.
Yeah, TBD, right? Great. Next question. Why might the over-inhibition be having a greater functional impact on individuals with SCI rather than over-excitation?
Yeah, good question. So when we talk about over-inhibition or over-excitation, those are very broad terms, and it very much depends on which neurons are over-inhibited versus over-excited. I think the three cases that I was talking about are all disinhibition. It's just who they're disinhibiting. So if they're disinhibiting neurons involved in pain processing, then you lead to excessive pain activation. If you disinhibit motor neurons, which innervate muscle, then that leads to excessive muscle firing, essentially, and spasticity. Mm-hmm. And if you disinhibit inhibitory neurons, so that's a lot of negatives layered on top of each other, but you get overdrive of inhibition. But it's still through the same mechanism, and my assumption is that they're all operating on different neurons. I think one of the nice pieces from Zhigang He's lab was they actually tried this. Mouse genetics means that you can like knock out or redeliver KCC2 only to certain neurons. And so they were able to show that like just giving it to excitatory neurons didn't do anything. So my assumption in this is that they're all three different pools of neurons. So it's not actually competing. Does that make sense?
Mm-hmm. Mm-hmm. Yeah, no, it does. Yeah, that makes a lot of sense. This relates to, we had a question come in that relates to an earlier point in the discussion, and that is there information available about what kind of exercise therapy, when we talk about exercise as a tool for rehab and regaining motor function, what kind of exercise therapy either has the most impact or, in the context of this talk, increases KCC2?
Great question. So all we know are studies that have been published. And I would say as an overarching theme, activity-based training seems to be way more beneficial than sort of general strength training. And the idea is you're having patterns of neurons that are getting activated together. And so that's automatically going to have a more collaborative effort and probably lead to better gain. I would say the linkage to KCC2 has not been tried in that many exercise models. So what you can do is take a rodent and make it run on a treadmill and show that you can increase KCC2 expression. But it's a little bit harder to compare exercise or rehab therapies. But yeah, I would say anything that generates patterns of activities is most likely to, but it has not been thoroughly explored.
Right, so we know what we know from what's been published, yeah. Okay, kind of a big picture question. One of the attendees has asked, is there a difference between ischemic versus traumatic injuries for this type of work? Is that known yet? Any information there?
So that is definitely not known yet. But what we do know is there are ischemic models of traumatic brain injury that lead to KCC2 downregulation. And so it wouldn't surprise me that there would be similar modality, but no one has actually looked at ischemic contributions to spinal cord injury in that way.
That's correlating from the central nervous system in general, right? A kind of another bigger picture question. Maybe talk a little bit about your experience in working in the field of developing treatments for spinal cord injury and using science, and kind of that interaction between, like, what's the current status out there with regards to pharmaceuticals supporting this work, that type of thing. Is it, like, where do you see this work coming from in the big picture of SCI research?
I think right now it's actually kind of an exciting time to be in spinal cord injury research. There are starting to be a number of biotech companies that are starting to actually translate some of these exciting findings. There's been a few in the past, so I don't want to discount that. But right now, I think there's a rapidly moving field. I would say that the biggest impediment to all of this moving forward is getting sort of venture capital on board. The field of spinal cord injury, I think, will benefit from getting an improved therapy. Basically, first out of the gate, if we have a success, then that sort of proves to some of the big funders of earlier stage just startups that this is reasonable to invest in. But that's not to say that there isn't general interest. You just need a startup who is committed to spinal cord injury, because most venture capitals will try to steer you in different directions.
Interesting. Yeah, interesting, the context. A question from a person with SCI. How do people with spinal cord injury find out if they're appropriate for clinical trials? Any like general advice?
Just general spinal cord injury.
Like if you're a person with a newer SCI, you know, how do you, I mean, I think we probably both comment on it, but from your perspective, like how would you know whether you qualify?
So there are some great tools out there. I believe one of my favorites is SCI Trial Finder, I believe, that has a way of sorting through clinical trials. I think the biggest caveat I would say is sign up for an actual clinical trial. Unfortunately, there are people that will peddle things that sound great, but if no one is overseeing it, they can't promise you. While I was at the Christopher and Dana Reeve Foundation, it's something like stem cells or something like exosomes are very vague terms. And so they can promise you that, but it could mean great things or it could mean bupkis. And so I think it's really important, if you're going to do a study, to do it with a center that's reputable and has some sort of oversight. That being said, there's a lot of cool studies going on. Depending on your injury severity or what the target is, sort of sorting through what is interesting near you is definitely worth doing, because it helps move the field forward.
Yeah, and I'd add to that, you know, akin to our activity here tonight, you know, going to the websites both nationally or in terms of your regional center for the spinal cord injury model systems is always a great, credible source of information. And usually, you know, local expertise available to help guide people through what may be a formal clinical trial versus something that you should proceed with a little more caution, or a little more skepticism, healthy skepticism.
Skepticism is always good. If a trial cannot explain to you why what they're doing is exciting and what the evidence is to support it, then like that's on them. Like they should be able to explain it to you.
Agreed. Okay, another big picture question. How far off do you think we are from clinical trials on humans?
It's a good question. I think I'm optimistic, so you should probably keep me on my word. I think it is important to sort of get all the ducks in the line first. And part of the reason that I like working where I'm working right now is everyone sort of is on the same page of, if I'm going to get to a no, that's okay. We have other options, but I want to find that out before we get anywhere close to humans. So make sure that what you're doing is actually going to work and that you know who it's going to work for. And then we can try. Optimistically, I'd like to say within the next couple of years. But that involves a lot of things lining up.
Yeah, for sure. Great. This is a question from one of our colleagues at Spaulding, Dr. Ryan Zielinski, asking if there are any radio tracers you know of where you can identify spinal KCC2 expression using PET MRI.
I don't, but that is a great question, and we should talk about that afterwards, Ryan.
Me too. Great. This is a really interesting question from an attendee. When obviously your talk was about addressing signal to noise ratio at the cellular level, but is there any work out there about addressing it at more of a macro level from the standpoint of like engineering techniques, anything like that? Kind of a broad question, but.
Well, one can argue. I think they're still trying to figure out sort of what neuromodulation is actually doing with either epidural stimulation or transcutaneous stimulation, but you could argue that that's essentially trying to get at a similar question. Mechanistically, we don't fully know how it's working, so we don't fully know how much it aligns. But I think that general stimulation to try to change excitability is one way to improve signal to noise. Other than that, most engineering is taking a bypass route. Like, even when I was in grad school, the cool thing was like, okay, well, let's be able to control a robot with your head. And you're like, that's cool, but also if you could get maybe like a little bit closer to the way biology works, it might be an easier path.
Yes.
And scalable.
Let's see. I have a series of questions from someone who's probably, I'm assuming, also involved in research and science, because it's fairly technical. So maybe I'll just ask a few of them so that we can make sure we get to everyone's questions. One of the questions is, does KCC2 enhancement need CL concentrations to increase in the body? Or how does the body approve or ignore this request? And maybe just for our audience, if you could explain that a little bit.
So KCC2 enhancement, I use that term because that can mean either making the pump more efficient or bringing more of the pump somewhere to do good. But either way, that will lead to less chloride within a neuron, which is the way a normal non-injured neuron functions. Within the body is a different question. One of the reasons that KCC2 is an enticing target, not just by us, but by others as well, in a number of different disorders, is that it is not expressed in the peripheral nervous system. It is not expressed in the kidney. The other pump that I referenced earlier in the talk, and KCC1, is. And so some of the side effects that might come along with targeting the other pump won't happen because of the spatial restriction of this pump. But yes, I think essentially the goal is to get chloride concentrations to decrease back to normal levels, the inverse of that question, by enhancing KCC2.
Okay, great. So another bigger picture question, kind of similar to what we discussed earlier versus traumatic versus ischemic injury, about whether there's any knowledge about this mechanism in other types of non-traumatic SCI like transverse myelitis, or is that still totally unknown?
I don't know the answer to that. I know that there was a couple studies looking at actually ALS, and that there is some transient change in KCC2. If I remember correctly, there is a decrease and then an increase. So it's not quite as straightforward as in spinal cord injury. But yeah, I'm not sure about that particular instance.
Okay, all right. So just want to tell the audience that we're starting to wind down on our questions. So if anyone has any late breaking questions or things they're curious about, please do write it into the chat now so that we can be sure to get it in while we still have some allotted time. Amanda, there's a question about other environmental factors in the spinal cord and impact on KCC2. Is that important, like things like pH or other aspects? of that cellular environment. You talked a little bit about the chloride ions, so maybe
Yeah.
expanding on that a little bit.
I can't tell you exactly what the relationship between pH is, but everything is kind of interrelated. Like KCC2, I'm talking about as a chloride pump, but it also pumps potassium. Like K is potassium, CC is 2 chlorides. And so when it's functioning, it's pumping one in and one out. It's pumping chloride out and potassium in. So pH is likely going to be related in some way, shape, or form. I just don't know what that actually looks like.
Sure. Makes sense. Big picture, basic question from someone with SCI. Can you use clinicaltrials.gov to search for trials in this area, or is there a better place to find that information?
Yeah, you can. You might get overwhelmed. Clinicaltrials.gov will also give you sort of like worldwide trials. So if you're going to do that, narrow by radius. So that, especially unless you want to be traveling internationally. But yeah, that will work. Just some of these other ways have like pruned a little bit more, and will allow you to filter more readily by things that you might know to search for, if that makes sense.
Yes, and Jenny Min from our one of our model systems key staff also put a link in the chat for scitrialsfinder.net, so folks can take a look at that. Noted in the chat if people haven't opened the chat yet, or you can find it there as a link to use for the future. All right, why don't we, I think if we could plan to do just a couple more questions, and then I think we'll be ready to move on for the evening. So again, any last questions, please be sure to put them into the Q&A or chat. This is a question. Why wouldn't you prioritize viral delivery over oral? Targeted effect to neurons of interest, upregulation of KCC2, and a lot less worry about off-target effects, like chemical binding to KCC1 and other reasons. So oral versus viral delivery.
Yeah, no, I was smiling when that question came in. That's sort of like an internal debate. There are essentially pros and cons to both. Oral delivery is the most straightforward way to humans, I would say, because there's always worry with viral delivery that you can't turn it off. Even if we're able to show, like in mice or rats, that we keep them around for like 3 years and nothing bad happens, there's still always going to be concern in humans if we can't have some sort of break on it. So the beauty of oral delivery is that you can stop taking it if something goes wrong. But like the question says, like, viral delivery allows you to target specific neurons. And so there is the ability, especially with some of the newer viruses, to make sure only certain neurons get it and not others. So that's definitely of interest. But yeah, it's guessing sort of which is more likely to succeed quicker. And that's not to rule out the other one.
Okay, that's really interesting. One question that's come in a couple times now, and maybe we'll take this as our penultimate question. So 2 more questions. This is related to KCC2, incomplete versus complete injury. I think you went into that in your talk, but maybe if you could just clarify, especially to our consumers, applicability to incomplete injuries.
Yeah, so I think it's important to distinguish between clinically complete and anatomically complete. And I know now with imaging technology, we can actually get at that question a little bit more. I'm actually a huge proponent of electrophysiology because I also think we should be doing that more in our human population, because then we can actually look at what remains and what doesn't. That being said, all of the spinal cord injury stuff that we've done with KCC2 to look at recovery of motor function has looked at a pretty severe injury that essentially the mice are not walking. And we get them to the point that they are generating locomotor rhythms. They're not, walking like we would normally want them to be walking, but they're doing much better. And so that suggests that there is essentially we need a minimal amount of tissue sparing. But what exactly that is actually something of great interest to us. And I think it will impact when we start talking about subject stratification to get into clinical trials, is that we want to know what is the minimum necessary to get functional benefit. And then that's not a clinical diagnosis, that's what is your anatomy actually doing?
Okay, that's great. So we're going to do one last question, and then I encourage everyone, if we weren't able to answer your question because of time restrictions, please feel free to email our spinal cord injury staff. Jenny Min is going to put her email into the chat, and we'll compile all the remaining questions that we weren't able to get to tonight, and send those to Dr. Zimmerman and try to connect you with answers. So last question for the evening. Kind of going back to kind of what people with spinal cord injury are hearing out there with regards to clinical trials versus other types of research and understanding the interaction from a consumer, would KCC2 be complementary to or alternative to stem cell therapy?
Yeah, I think that one of the nice things about a KCC2 treatment is there is no reason that it wouldn't work in combination with a lot of other things. Essentially, KCC2 treatment is not working on regeneration.
Mm-hmm.
It is working on getting more out of what remains. So any sort of therapeutic that works on regeneration might have a additive benefit. And some of the other research that Axonis is working on is actually curious to see whether that might happen down the line.
Mm-hmm. Wonderful. Okay, thanks. Well, also coming in on the chat were many thanks from both the audience, Dr. Zimmerman, for your fantastic lecture and for sticking with us for this really interactive and interesting Q&A. And we from the Spaulding New England Regional Spinal Cord Injury Model Systems also thank you so much for your time and bringing your expertise to our Knowledge in Motion lecture series. So thanks you, Dr. Zimmerman. A reminder to everyone to please fill out your surveys. The link was posted in the chat throughout the presentation. When you close Zoom, you should be prompted to an online survey, and we'll also email that out to anyone who registered for this lecture. We'd love to hear your feedback and ideas for future topics for the Knowledge in Motion lecture series. So thank you everyone for joining us this evening. And we hope everyone has a great rest of the evening. Stay safe out there. Our next Knowledge in Motion lecture will likely be in November. More information to come on that as we begin the planning. Thank you again, everyone. Have a great night.
Thank you.